Hi, this is Dr. LeBlanc again from the Department of Biology at Merrimack College. In our previous lecture, we learned that many members of the scientific community contributed to this discovery that DNA was the genetic material. Likewise, in this lecture, we're going to learn that the discovery of the structure of DNA, was the product of the discovery of many members of the scientific community. Our learning objectives for this lecture are to understand the components of DNA and RNA, know the major contributors to the discovery of the structure of DNA, know the features of the DNA double helix, and to memorize the basic chemical structure of the DNA molecule. Now, why do I want my students to memorize a chemical structure? It's because knowledge is power, and if you have this basic chemical structure of a DNA molecule memorized, you'll be able to understand many other concepts that we will cover in subsequent lectures. Here's the iconic picture of DNA. You notice that it has two strands linked together here by hydrogen bonding between the bases, and we're going to talk a lot more about this structure. So the discovery of the structure of DNA was the work of the scientific community, and many scientists contributed. Linus Pauling, who worked primarily with protein structure, proposed the alpha helical structure. Rosalind Franklin and Maurice Wilkin took the actual X-ray diffraction pictures of the DNA molecule that helped Watson and Crick solve the mathematical problem of what the structure of DNA was. And Erwin Chargaff did a very simple experiment that proved to be provide one of the key elements of the structure of DNA. Here's Linus Pauling holding a model of a protein molecule. This alpha helix structure shows that proteins can form a three-dimensional structure in a helical form. And the way that they can form this helix is by hydrogen bonding between the backbone of the protein molecule and its side chains. Two of the most important members of the team that solved the DNA structure were Rosalind Franklin and Maurice Wilkins. Right here, this is Maurice, and he's standing next to the structure of DNA. They took X-ray diffraction pictures of the DNA molecule to show that the most probable structure was a double helix, not a single helix, but a double helix. And that was also a very key point of information needed to solve its structure. How do you take an X-ray picture, X-ray diffraction picture? You take a beam of electrons and you bombard a crystallized form of your molecule, and as the electrons bounce off the crystal, they form a diffraction pattern, a scatter pattern on a piece of photographic film. And using mathematical analysis, you can determine what the structure of the molecule is based on that pattern of diffraction. This is a very famous photo 51, which was taken by Raymond Gosling, who was a student of Rosalind Franklin. There's a very famous or infamous story behind this photograph. Maurice Wilkins went into Rosalind Franklin's desk without her knowing it at all or her approval, took the photograph out of her laboratory notebook and showed it to James Watson, who, when he saw it, thought that it proved that DNA, the structure of DNA was a double helix because of this X-ray diffraction pattern. This is a very famous controversy in science, and you can read a lot more about it. There's even a play that was written about this, so a lot of drama behind the scientific discovery of the structure of DNA. Erwin Chargaff in 1950 published this table of data. He asked a very, very simple question that had a very profound answer. He asked how much adenine, thymine, guanine, and cytosine, the bases of DNA, were there in an organism. So he looked at E. coli and said, there's about 26% adenine, about 24% thymine, about 25% guanine, 25% cytosine. Within his experimental error, these numbers were all equivalent. So he said, okay, there's an equal amount of adenine, thymine, guanine, and cytosine, which would have been a very uninteresting answer or uninformative answer if he had stopped there. Then he looked at Streptococcus pneumoniae. Remember Griffith's and Avery McCarty and McLeod's experiment? When he looked at strep, he found there was approximately 30% adenine and 30% thymine, but only 20% guanine and 20% cytosine. So he continued to march through organisms, and in every case, he found that the amount of adenine was approximately the same as the amount of thymine, and the amount of guanine was approximately the same amount of cytosine. Looking at the chemical structure of these different molecules, these different bases, he proposed that adenine and thymine could hydrogen bond together, and guanine and cytosine could also hydrogen bond together. So that led a very important piece of information to solving the structure of the DNA molecule. So Watson and Crick designed models and had the machine shop make them for them and started to fit them together. And finally, with Chargaff's data and Rosalind Franklin's data, built this model of DNA. In April 1953, legend has it, April 25th, 1953, they walked into the local pub, being proper Englishmen, and raised a pint and said, "We've solved it. We found the structure, or we discovered what the structure of DNA was." On April 25th, 2003, the Human Genome Project announced the first draft of the human genome. So April 25th now is known as DNA Day. Okay, so there's a hierarchical order to the structure of DNA. The nucleotides are the individual building blocks. They're the repeating units. They're the A, the G's, the C's, and the T's. They get strung together to form a single strand, which then can hydrogen bond to another single strand and form what we know as the iconic structure of DNA, the double helix. What people sometimes forget is that this single, the double-stranded molecule can also fold up on itself and form a three-dimensional structure. And when we learn about the structure of chromosomes, we'll understand the complex manner in which the DNA double strand can fold up. So here's the nucleotide structure. It has a base, and it has a sugar. This is the sugar part, and a phosphate group. Okay, this particular sugar is deoxyribose. Okay, why is it deoxy? Why is it missing an oxygen? Well, ribose would normally have an oxygen here as well. Okay, but deoxyribose doesn't have this oxygen, so it's deoxyribose. The carbon atoms of the sugar moiety are numbered from the point of attachment of the base. So the first carbon is the carbon to which the base is attached, the A, the T, the G, or the C. A, T, G, or C. Okay, and then as we go around the carbon, the second carbon is here. This is the 3' carbon, the 4' carbon, the 5' carbon. They have this little prime attached to it to denote the difference between the carbons of the sugar and the carbons of the base. Notice in particular that this carbon in the last position of the sugar, the 5th or 5' carbon, is actually not in the ring portion of the carbon. It's dangling off. To that is attached a phosphate group. It's important to notice that the 5' carbon of the sugar and the 3' carbon of the sugar are the two carbons that are referred to when we talk about a strand of DNA having a 5' end or a 3' end. And we'll come back to this often over and over again as we talk about the structure of DNA in more detail. 4 and our jumping 5. Okay, so when we attach all these nucleotides together in a long strand of DNA, notice we have our 3' end here. Here's our 3' end, free 3' end with a hydroxyl group on it, and our free 5' end here with a phosphate group on it. These phosphodiester linkages are between the 5' carbon of one sugar and the 3' carbon of the next sugar. We always say that there is a 5', 3' phosphodiester linkage. Okay, you have to remember when you're drawing this that the 5' carbon is not part of the ring structure of the ribose, okay, but the 3' carbon is. So here's our phosphate group. Remember, phosphorus has 5 valence electrons, so we need to account for that when we draw the structure. A phosphate group has 4 oxygens, and this one extra electron, of course, is really running all around. Here, it's not stationary, but we have to draw it and attach to one of the oxygens. And these phosphates on the edge of the side of the backbone make this DNA molecule a very negative charge. The bases, on the other hand, are hydrophobic, so that when the two strands hook up with each other by hydrogen bonding, all of the hydrophobic bases are nestled in the center of the DNA helix, and the sugar phosphate backbone is exposed to the aqueous solution, the water. Okay, so here we have our double helix, classic iconic structure. This is a ball and stick diagram. Here's a space-filling model. Notice, first of all, that the bases form really steps of a spiral staircase. They're pretty planar. They're almost completely perpendicular to the axis. Okay, here's the axis. So they're almost completely perpendicular to the axis, the bases. Okay. And like I said before, they are hydrophobic, and so they're encased in the middle of the double helix. As DNA twists around itself, it forms two types of grooves, a major groove and a minor groove. And this is a very important feature to look at when you're looking at DNA models, actual DNA models. If they're very accurate, they will have both a major groove, which is a wide, deep groove, and a minor groove, which is a narrow, shallow groove. In the major groove, the bases are exposed, and so if you're a protein and you want to interact with specific sequences of bases, you are going to bind in the major groove. Okay, and remember, our sugar phosphate backbone is sticking out where it's exposed to the aqueous solution. Most DNA forms this B structure, where it's approximately 10 bases per turn, and you have a nice deep major groove here and a shallow minor groove. And the bases are, again, perpendicular to the axis of the molecule. There have been reports in the literature of zDNA. This is basically an experimental test tube version of DNA. It is a opposite helix instead of a left-handed helix. This is a right-handed helix, and you notice that the bases are not perpendicular to the axis, but they're on a slant. It's wider as well. And like I said, it will pop up in the literature. You may encounter it in your readings, but it's largely an artifact of experimentation. Okay, and this is a space-filling model that shows that the backbone also of zDNA is in a zigzag pattern, whereas in bDNA it's nice and smooth. It goes all the way around like that. Okay, so here's our flattened model of a DNA molecule. We have our double-stranded molecule, and now it's complemented by hydrogen bonding between the bases. Here's where the hydrogen bonds all form. Notice that there are two patterns of hydrogen bondings that form between the AT bases, 2 hydrogen bonds, and the GC bases, the 3 hydrogen bonds. And many different processes involving DNA replication and transcription exploit these what seems like very minor differences between the number of hydrogen bonds between G and C and A and T. Notice also that the space between the sugars here does not vary along the entire DNA molecule, and that is because a GC pair has approximately the same width as an AT pair. Here are some of the important dimensions of what a DNA strand is. There are approximately 10 nucleotides in each turn, and they're anti-parallel to each other. That's a very important point. They're anti-parallel to each other, so that the 5' end of one of the strands is opposite the 3' end of another. And if you look at the sugars here, notice that the oxygen, I like to think of these as an arrow, the oxygen is pointing up here, and the oxygen is pointing down here. Anti-parallel, that's a very important feature. Okay, so let's look at each of the components individually. We have our phosphate group, I already talked about that. We have our sugars, and I already talked about deoxyribose. This is ribose, of course. Why is it ribose? Because it's not deoxyribose. It has its oxygen on the 2' carbon. It has a hydroxyl group, whereas deoxyribose has no oxygen here. Losing this oxygen makes this a much more stable molecule. This oxygen dangling out in space like this makes RNA a very reactive labile molecule, which means it can degrade easily. What about the bases? So here are 5 different bases that we can find in nucleic acids. These 4 are found in DNA. This is found in RNA, uracil. Uracil takes the place of thymine in RNA. Okay, but RNA also has cytosine, guanine, and adenine. Okay, 2 different types of bases, purines and pyrimidines. And the way I like to remember it is the short name has the big bases, 2 ring structures, and the big name has the smaller bases with a single ring structure in them. Here's a molecule of ribose, and the nomenclature for just the sugar and the base is adenosine. If you add a single phosphate group, it's adenosine monophosphate. 2 phosphates, it's adenosine diphosphates. And 3 phosphates, it's adenosine triphosphate, also known as ATP. If you want to be a powerful genetic student of genetics, you should memorize this structure. If you memorize the phosphodiester linkage between the bases, you will understand that one of the strands, a single strand of DNA, has a 3' end, and what that actually means. It means that the 3' carbon is free. And at 5' end, you will remember that the 5' end and the 3' end are anti-parallel. The two strands are anti-parallel. You will remember that an adenine pairs with thymine and has 2 hydrogen bonds, and a cytosine pairs with guanine and it has 3 hydrogen bonds. You'll also remember that the fact that these two strands are anti-parallel means that one oxygen is pointing in one direction and the other oxygen is pointing in the other direction of the 2 riboses. One trick that I often use when I'm learning how to draw this or remembering how to draw this is that I'll draw one strand and then I'll flip the paper around and just draw the other strand. Okay, so in summary, we had an overview of the DNA structure. We've reviewed what the key experimental data was that contributed to the discovery that DNA is a double helix. We learned that there are different types of nucleic acids, what their structure of the nucleotide is, the 4 levels of complexity of the DNA, a few key features of the DNA strand, such as the major and minor grooves, anti-parallel, the fact that the bases are perpendicular to the axis of the DNA, all of these things here. There's a hydrophilic backbone and a hydrophobic core. There's B and Z DNA, and a very important structure to memorize.